Hi there. Welcome to our mini lessons for chapter 13, which is gene expression and protein synthesis. So this chapter kind of helps to culminate a lot of what we've done up to this point with cell structure and protein structure and DNA and just putting all the pieces together. So this is another really good culmination chapter. So when we talk about gene expression, we have to remember what a gene is. So a chromosome, we talked about genes like when we're talking about genetics, your dominant and recessive alleles, different versions of a gene. So what is a gene? So if you remember, we have our chromosomes. Chromosomes are just pieces of DNA. So this is a piece of DNA. We just got done covering chapter 12, the structure of DNA with all the A's, T's, C's, and G's, right? So that's what a chromosome is. It's just one piece of DNA. And then a gene is a particular sequence, just a shorter piece, a section of that particular DNA. So let's say this DNA right here, just that region of all the A's, T's, C's, and G's, that's just right there, that's going to be one gene, and that's going to code for a particular trait. This is a gene, and it codes for traits. So all the stuff that we're looking at back in our genetics chapters 11 and 16, the trait might be taco tongue, right? So that's the gene. There is a gene on your chromosomes that has the instructions to either allow you to do taco tongue or no taco tongue. Those are the two different alleles, the different versions of that gene, or freckles on your face, or the widow's peak, or the hitchhiker's thumb, or the knuckle hair, or the attached earlobes, whatever the trait is. For a lot of traits, not all, because we saw all the different kinds of unique inheritance patterns in chapter 11, like polygenetic or epigenetic or not epigenetic, epistasis, all those different kind of more complex inheritance patterns. But for the most part, the pattern holds true that there's a section of DNA called a gene, and it codes for a particular characteristic or trait. The genotype is the version of the gene that you have, dominant, recessive, codominant, and then the expression of that trait is your phenotype. So we're kind of culminating some of our genetics understanding to the actual molecular instructions for how that genetics works. So before this was understood, again, kind of going back a little bit in history, people understood, they were having a good understanding of DNA thanks to Watson and Crick and the structure of DNA. But the understanding of how the instructions are in the DNA actually made stuff, like what's the connection between the DNA and a cell product? And in this case, we're specifically going to be talking about proteins. So a gene, so let's say maybe this one down here, kind of I'm coloring green. This is a gene, and instead of saying codes for a trait, we're going to be a little bit more specific and say codes for a protein. So we can see traits are basically the expression of particular protein. I said traits. I meant to write proteins, not traits. So traits are like your proteins. If you are able to express a particular protein, that's what allows you to have the widow's peak and the taco tongue and the attached earlobes, all those traits that we've been talking about. So taking a look at the historical connection of who kind of discovered this relationship, we have Linus Pauling and Ingram and this idea of hemoglobin. So hemoglobin is the protein that is inside of your red blood cells. Its job is to carry oxygen. So it is made up of just kind of 4 squiggly subunits. They're called alpha and beta. You have 2 alpha and 2 beta subunits. And right in the middle, right in the very middle of these hemoglobin subunits, the alpha and beta subunits, there's an iron group. It's called the hemoglobin. And then attracted to that iron is where you're going to be able to carry oxygen. So the job of hemoglobin is to carry, each molecule of hemoglobin can carry 4 molecules of oxygen. And its job is to pick up all the oxygen from your lungs, take it out to your tissues, drop off the oxygen. So then your tissues can do aerobic cellular respiration, right? We know all about that now. Now, in an individual who has sickle cell, well, let me back up. Your red blood cells are jam-packed full of hemoglobin, like 250 million molecules of hemoglobin or something. It's an insane amount. So your red blood cells are jam-packed full of hemoglobin. Okay. So inside you have, 200 plus million molecules of hemoglobin inside. And your red blood cells are shaped very specifically, like we can see in this picture here. They're what's called a biconcave disc. So it's like a donut that's not punched all the way through. It's got little dents instead of an actual donut hole. Now, structure and function plays a big role in the shape of the red blood cell. So the little dent on either side, the indentation helps to increase surface area, which makes gas exchange, the oxygen and the CO2 that the blood cells carry, a lot more efficient. So that's good. And what it also does is it's really flexible. It helps to bend and twist itself through tiny, tiny little capillaries. And also, what's not mentioned here, which isn't directly involved with hemoglobin, is red blood cells lack a nucleus. They have a nucleus when they're born. In their maturation process, they eventually lose it. So they are technically eukaryotic cells at the beginning, but then they're modified to just serve this whole purpose of carrying hemoglobin, which allows our blood to carry oxygen really, really well. So in normal red blood cells, with a normal hemoglobin protein, so here's our particular sequence. Now we got to do a little bit of reaching back to chapter 3 when we were learning about protein structure, right? So proteins are big long chains of amino acids, right? So this was primary, and then they fold either into a helix or a pleated sheet, that was secondary, and then they kind of get globular, right? And that was tertiary. So what we're going to be looking at here in this diagram is the primary sequence. So for a particular region in that big, huge hemoglobin protein chain, this is the correct order of those amino acids: valine, histidine, leucine, threonine, proline, glutamic acid, blah, blah, blah. And it's going to go on in either direction. That's a particular location. I think it's glutamic acid 563 or something like that. So when the amino acids are in this particular order, that's a good folding hemoglobin. It's going to get those 4 subunits, the alpha and beta subunits are folding just right. When oxygen is there, when oxygen is absent, they are properly folded. Now in a mutation, take a look at the sequence of amino acids in a sickle cell hemoglobin molecule. Valine, histidine, leucine, threonine, proline, valine instead of glutamic acid, glutamic acid. So if I'm going to draw on my little primary structure over here, so normal, 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 abnormal, normal, normal. So with the wrong amino acid in that primary sequence, it is going to affect the secondary and the tertiary folding. And so what you get is a misshapen hemoglobin molecule. Now, it's not just one, it is every single hemoglobin molecule in every single red blood cell. So for individuals with sickle cell anemia, it's one mistake, changing one amino acid, changes the structure of the entire protein, which changes the function of the entire protein. So here's that structure and function relationship again. So what happens with sickle cell patients is when they have low oxygen concentrations in their lungs or in their tissues, like when they just get up to walk across the room, their muscles are working, they're taking up the oxygen. Instead of having these nice round cells, what ends up happening is they turn into these elongated, what's called sickle shape, kind of like a banana. So these are red blood cells that don't move through the capillaries very well. They don't hold oxygen very well. And so that's the problem. People with sickle cell anemia do not carry oxygen to their tissues very well. And these red blood cells can get stuck in your capillaries, which can be very painful. It can cause tissue damage, and eventually it can cause death. So what's the mistake? So we have a valine instead of a glutamic acid, but where are the instructions coming from that give us that characteristic? Well, we're going to be learning in this chapter, it is the sequence of those A's, T's, C's, and G's that give our cells the instructions for which amino acid goes where in a protein structure. So let's take a look at this right here, this 3-letter sequence. So in your normal DNA in a proper hemoglobin, that would read, I'm just going to read on the bottom line, GAG. So GAG, that particular three-letter combination codes for glutamic acid. See how that is different in the mutated form. Instead of GAG, it's GTG. Now, GTG does not code for glutamic acid. In this case, it codes for valine, which is the wrong amino acid, and that's the mutation. So whenever we talk about mutations, it's always going to be a change in the DNA sequence, but because it's a change in the DNA sequence, that is going to have a downstream effect on the proteins because DNA codes for proteins. In the context of this class, that's what we're going to be going with. So we have what's called the central dogma, and I don't think your text uses that terminology anymore, but I like it because it's kind of ingrained in history. The idea of central dogma is that the information, the instructions, is coded for in a molecule of DNA. That DNA is then copied in a special process called transcription into a very similar type of molecule called RNA. So DNA was deoxyribonucleic acid, RNA is just ribonucleic acid. And then through another special process called translation, that RNA is converted into a protein product. Okay, so this is the central dogma, DNA to RNA, RNA to proteins. It is the path of gene expression. So again, here's my gene. I'm just going to highlight it with the blue. So let's say that is the sequence of DNA that when transcribed will make a short little piece of RNA. And then when translated, that short little piece of RNA will make a functional protein. That's your product. Okay. So what is RNA and how is it compared to DNA? Well, I have a slice of RNA over here on the side, and if you'll notice, it looks very similar to a molecule of DNA. It's got the nucleotide, so I'll just circle one of those nucleotides here. We have a phosphate group, we have a sugar, and we have a nitrogenous base. So we have all of the three parts, but there are some differences, right? So one of the differences, take a look at the sugar. It's ribose sugar. It's not deoxyribose, it's ribose. And really, it's just this OH group. So that one I just highlighted, that's missing on the DNA. So if you go back and take a look at the deoxyribose sugar in a DNA molecule, you'll notice it just has an H there instead of an OH. So it's deoxyribose. It's missing the oxy part. So ribose is just a regular ribose sugar. The phosphate groups are the same. The ribose is a different sugar. And if you'll notice, we've got a new base. We have the same ones, guanine, cytosine, adenine, but what are we missing? Thymine is replaced by uracil. So uracil is a difference between RNA and DNA. DNA has those four bases, A's, T's, C's, and G's, whereas RNA has A's, U's, C's, and G's. Okay. Another difference between DNA and RNA is RNA is single-stranded, kind of like what we see here. It's not a double helix. It's a single sugar phosphate backbone with the bases sticking off, the A's, U's, C's, and G's, and that's it. Another difference is RNA is a lot smaller. So your RNA is only going to be as long as your gene is long. And your DNA is huge. Like each chromosome is hundreds of thousands of base pairs long, where a piece of RNA might only be like 1,000 base pairs long. Just the information needed to code for that one protein. And you could have hundreds of genes on a single chromosome. So if you have like a chromosome, oops, like this, right? If this is your chromosome, your piece of DNA, not to scale, right? You could have, that could be a gene. That could be a gene. You know, these different regions, these banding patterns, sometimes you see DNA with like banding patterns. Each one of these are representing a different gene that's coding for a different protein. So your one chromosome could have multiple genes on it, and each one of those would get transcribed into a piece of RNA that's special for that protein and then translated into that protein. All right, so this is just the introduction, a little bit of history, kind of the rationale behind why it's so important to have DNA be copied correctly, be transcribed correctly, and then translated correctly, because it will ultimately result in either a normal functional protein or an abnormal non-functional protein. And almost don't want to say almost all, but a lot of human diseases are caused by mutations in the DNA that produce non-functioning or malfunctioning proteins. Cystic fibrosis, you're doing your genetics case study, or you did your genetics case study on cystic fibrosis. That one cystic fibrosis gene, there's a mutation. There's a couple mutations you had to look up and research, right? So those are the mutations in the DNA that lead to a malfunctioning protein for ion transport, and that leads to the disease of cystic fibrosis. So it's all in the genes, if you will, that code for these particular protein products. Okay, so that's our introduction, and I will see you next time for moving on deeper into gene expression and protein synthesis.